Good afternoon, students. Today we are going to discuss a new topic. Yesterday we have already discussed about how transcription processes occur in both eukaryotic and prokaryotic in shortly. In this session, we will discuss in detail about the process of transcription, how occurs in the prokaryotic and also eukaryotic. Next slide, please. In this session, we will discuss some points, in which first point is the types of RNA and the process of transcription, and how transcription process occur in the prokaryotic cell and also eukaryotic cell. So we will start with this point, type of RNA and the process of transcription. In last lecture, we have already completed how transcription unit work and which components which is included in the transcription unit. You must know there are three components which is present in the transcription unit, in which first is the promoter, second one is the structural gene, and last one is the terminator or termination in. Okay, so with this, with the help of this transcription unit, it is very easy to understand the types of RNA and also the process of transcription and role of the RNA in the process of transcription. Okay, so listen carefully. In bacteria, there are three major types of RNAs. We know that bacteria, which is considered as the prokaryotic cell because it is a unicellular and which falls in the kingdom Monera. In bacteria, there are three major types of the RNAs. mRNA means messenger RNA, tRNA means transfer RNA, and last one is the rRNA means ribosomal RNA. All three RNAs are needed to synthesize a protein in the cell. Means what? In the process of synthesis of protein synthesis process, in the process of protein synthesis, all three RNAs are very essential to complete the process of protein synthesis. Okay, the mRNA provides the template. Means what? The mRNA, which piece of mRNA included the genetic information which is coded by the DNA. tRNA brings the amino acids. Means what? tRNA means what? The transfer RNA. All these processes are occur inside the cell. Means what? The transcription process is occur inside the nucleus. That's why firstly mRNA acts as a template and tRNA brings amino acids and reads the genetic code. Means what? tRNA brings the amino acids. There are in nature 21 amino acids are present. They identified and bring the amino acids and reads its genetic code. And rRNA, it is the third type of the RNA. rRNA means what? The ribosomal RNA, which is present on the endoplasmic reticulum. rRNA plays structural and catalytic role during the transcription, translation. Next process is the translation. Means what? The mRNA is converted into the protein. Means what? Here protein synthesis process is completed. Means what? It is the another type, but we only focus on the process of transcription. Next slide, please. There is a single DNA dependent RNA. Yesterday we are completed the discussion about how a transcription process occur and what is the role of DNA dependent RNA polymerase in the process of transcription. There is a single DNA dependent RNA polymerase that catalyzes transcription of all types of the RNA in bacteria. Means what? In case of prokaryotic cell, means what? The example of prokaryotic cell is the bacteria, and in this case what happen? How transcription processes occur? RNA polymerase is the enzyme which enhance or which initiate the process of transcription when it is binded with the promoter, and promoter is present in the structural unit of transcription unit. Okay, RNA polymerase binds to the promoter and initiate transcription initiation process. And you also know that the transcription process included three steps, in which first is the initiation, second one is the elongation, and third one is the termination. So here first step, initiation which is started with the RNA polymerase, which is the enzyme which is binded with the promoter of a transcription unit. It uses nucleoside triphosphate means or nucleotide as a substrate, and you must know every when any enzyme is going to react, it is very important of the presence of substrate there, and here substrate is the nucleoside triphosphate as a substrate, and polymerases in a template depends fashion following the rule of complementary. So means what? In the process of transcription, it also follow the rule of complementarity, means of the base pairing. You must know adenine is binded with thymine, and thymine is binded with guanine. In case of DNA, in case of RNA, thymine is replaced by the uracil. Next slide, please. See here, transcription in bacteria. How transcription process occur in the bacteria means what? How transcription process in prokaryotic cell occur. In the process of transcription, there are three steps are there, in which first is the initiation, second one is the elongation, and third one is the termination, which is represented here in the diagrammatic view. Okay, so this is the DNA fragment, and which shows the how transcription process occurs in the bacteria, means or the prokaryotic cells. And in last period we have already discussed about the cystone, types of the cystone, monocystonic, polycystonic, and here we focus on the bacteria. That's why in the bacteria which kind of the cystones are present? Polycystonic nature of the structural gene is present. So look at here, this is the DNA fragment, and you must know transcription processes start with the help of DNA dependent RNA polymerase, and which binds with the promoter of structural transcription unit. Means what? The RNA polymerase is bind with the promoter, and one factor is also needed to initiate the transcription process, and this is the sigma factor. Means what? Sigma factor is very important to initiate, to start the process of transcription. And when RNA polymerase, this is the enzyme, when RNA polymerase is binded with the another factor or sigma factor, which form the holoenzyme, and holoenzyme is also worked here to initiate the process of transcription. Means what? The promoter, RNA polymerase, and sigma factor start the process of transcription in the bacteria. Okay, so this portion is the promoter, and middle portion shows the elongation process, and last one is the last portion is represented by the terminator. Okay, so this is the initiation when RNA polymerase, sigma factor, promoter is bind here. Then elongation process in which RNA is formed, and RNA in the RNA fragment or in the RNA piece, the genetic information is coded, and when the elongation process is completed, then the RNA and another or terminator factor is act on the RNA, DNA dependent RNA polymerase, and the RNA polymerase is removed here from this sequence, and with this step is termed as the termination of transcription in the bacteria. So remember it very clearly, because it is very important diagram for examination point of view. Next. So look at here, it somehow also facilitates opening of the helix and continuous elongation. Means what? In the previous diagram we observe three steps: initiation, elongation, and termination. In which in this line the point is cleared, initiation point is cleared, so that which continued the elongation, and last one is the termination. Okay, next slide please. So this is all about how transcription process is occur in the bacterial or eukaryotic cell. But what happened in the process of transcription in eukaryotic? Because transcription process is very difficult in the eukaryotics, because the eukaryotes having well defined nucleus. Okay, that's why it is very difficult, but it is similar to the prokaryotics, but some two complexities are there in the process of transcription in eukaryotics. So we are going to discuss in details. See here, this is the DNA fragment. Again, it is proceeding to three steps, in which the initiation, elongation and termination is there. And in the eukaryote, which kind of the structure is present? Monocystonic, means what? The DNA segment is coded by the polynucleotide or protein, which is termed as the cystone. Means what? Here one piece or one kind of the piece of coding information from the DNA is present here. That's why it is the monocystonic unit. Okay, so monocystonic unit, or this piece of RNA having different two kinds of the interrupted parts here, in which yesterday we have already discussed about it. Exons and introns are there. This yellow portion is presented as the exons, and red portion is presented as the introns. Exons are the functional parts and the introns are the non-functional parts, because it is not coded, and the exons are the coded for different sequences. Okay, that's why it is very important to know about the exons and the introns. In the RNA splicing, what happened? From this sequence, the non-functional, means what? Non-coding parts are removed, means introns are removed, and functional and coding part is continued, and they form the messenger RNA, mRNA, messenger RNA, in which the genetic material or genetic information is coded, and which is proceed for the process of translation, and it is a very important process for the synthesis of the new kind of the proteins. Okay, so this is all over about the transcription in eukaryotics, in which three processes are, or three steps are there, in which the initiation, then elongation and termination, as like the prokaryotic cells. Then next, in eukaryotics there are two additional complexities. So I mentioned earlier, there are two complexities in the eukaryotic cells, in which in eukaryotic there are two additional complexities are there. There are at least three RNA polymerases in the nucleus. Means what? In the nucleus, because you must know, you know that the eukaryotic cell having a well developed nucleus. That's why these two complexities are there. In the nucleus there are at least three RNA polymerases in the nucleus. Inside the nucleus there are three kinds of the RNA polymerases are there, in addition to the RNA polymerase. DNA dependent RNA polymerase is also present, but other than this DNA dependent RNA polymerase, another three RNA polymerase are required to complete the process of transcription in case of eukaryotic cell, because they have the well developed nucleus. Okay, so there is a clear-cut division of the labor, and these additional three RNA polymerases plays very important role differently, and they divide their work. That's why here there is a clear-cut division of the labor. The RNA polymerases first, means what? Three RNA polymerases are divided into three parts, in which first is the first RNA polymerase first, RNA polymerase second, and RNA polymerase third. So we will discuss point wise. First we take the RNA polymerase first in the process of transcription. So the RNA polymerase first transcribed RNAs 20 years and 80 years and 58 years, whereas the RNA polymerase third, means what? First RNA polymerase first role is different than the RNA polymerase third. is responsible for the transcription of tRNA. Transfer RNA is transcribed by the RNA polymerase third. And sRNA, sRNA means what? The small nuclei. It is very important. You prepare all short forms and their long forms for the examination point of views, because many times in multiple choice questions this question is asked. Okay, so sRNA means what? The small nuclear RNAs. The RNA polymerase second means what? We have already covered the RNA polymerase first, role of RNA polymerase first, role of RNase second, polymerase second, third, and we are going to discuss about the role of RNA polymerase second. The RNA polymerase second transcribes the precursor of mRNA. The heterogeneous nuclear RNA means what? It plays an important role in the formation of heterogeneous nuclear RNA. Okay, so all those three RNA polymerase, including RNA polymerase first, RNA polymerase second, and RNA polymerase third, play different roles in the process of unique process of the transcription in eukaryotes. Next slide, please. And this is the first complexity, and the second complexity. The second complexity is that the primary transcript contain both the exons and the introns and are non-functional. Hence, it is the subjected to process called as splicing. Means what? In first case, you already observed that the piece of RNA which is transcribed during the process of transcription, they are having both the pieces of exons and introns, and when we are remove the introns, which is non-functional or non-coded, and they form the mRNA, means what? Messenger RNA, which is the totally coded by the genetic information. And this process is called as the splicing process, defined as the splicing. Heterogeneous nuclear RNA undergoes additional processing called as the capping and tailing. Okay, capping an unusual nucleotide methylene gonose and triphosphate is added to the 5' of end of the heterogeneous nuclear RNA. In tailing, adenylate residues 200 to 300 are added at 3' end of the sequence in the template independent manner. Next slide, please. Okay, so it is fully processed HN RNA, and HN RNA means what? Heterogeneous nuclear RNA. Okay, now called the mRNA, and that's the final product is the mRNA, which is having only exons, not introns, because introns are removed by the process of splicing, and the mRNA piece is the totally coded by the genetic information, and that's why here writing it is the fully processed HN RNA, heterogeneous nuclear RNA, now called as the mRNA, means or the messenger RNA. And this messenger RNA, this piece of the coded information of the genetic information, this is used for the next process. This is the translation process, and mRNA is very important. And in we already discussed in the process of central dogma, the DNA is converted into mRNA, and mRNA is converted, or they produce the protein, and this process, this is the next process of the discussion. This is the translation. So in case of transcription, only DNA is converted, or from the DNA sequence we produce the mRNA, and this is the limited to the transcription process. Okay, that is the transported out of the nucleus for translocation. Means what? The first in the central dogma, the first process is the DNA replication, and when DNA is converted into the mRNA, this process is called as the transcription. And transcription in process of third one is the process of translation. In the central dogma, translation is the conversion of mRNA into the protein. This is the final synthesis of protein, and it is a very important process in the term of the genetics. Okay, next slide please. Okay, thank you. I hope you are understood the process of transcription both in prokaryotic cell and eukaryotic cell. Thank you very much.